Hello everyone. Welcome to lecture 4 on plant growth and development. So in last lecture we discussed about different plant growth regulators. So we'll just have a quick revision about their functions. So in the last lecture we discussed about auxin, gibberellins, cytokinins, abscisic acid and ethylene. So ethylene was the only gaseous plant growth regulator. So in the last class, we discussed PGRs, that is plant growth regulators that are signaling molecules in plants. So auxin, auxin also known as indole acetic acid, IAA. Auxins, which are the growth promoting hormone, they are growth promoting hormone. It promotes the growth. So they are growth promoting hormone. They help in cell elongation. They play an important role in apical dominance. Apical dominance, which is the bending of shoot towards the light, and tropism. Phototropism is the effect of this, the presence of auxin, and also they play an important role in fruit development. They are growth promoting hormones. The second one is gibberellin, which is also again a growth promoting hormone. So it also promotes the growth. What's growth? So gibberellins, as we remember, the discovery was based on the elongation of the stem. So the main function to remember for this gibberellin is stem elongation. Like apical dominance is the major function of auxin. Stem elongation is majorly related to gibberellin. It is also related to flowering and germination. So gibberellin is mainly related to stem elongation, flowering, and germination. Cytokinin is again a plant growth promoter, and it mainly helps, as the name suggests, cytokinins. That is the one that help in cell division. So they mainly help in cell division, and differentiation. Cell differentiation. Also, it suppresses apical dominance. It suppress apical dominance. So it is antagonistic to auxin in this way that it suppresses apical dominance. Apical dominance is promoted by auxins, but cytokinins suppress apical dominance and senescence. Now, ABA is abscisic acid is the stress hormone. The stress hormone, and it is a growth inhibiting hormone. Growth inhibitor. So, since it's a growth inhibitor, it will cause dormancy. It will not allow the plant to grow. Germination will be prevented by the dormancy. So, it causes dormancy, and it also causes stomatal closure during the water stress. So, it prevents the plant from the stress. So, water stress is prevented by the stomatal closure, which is caused by the ABA. That is the abscisic acid, which is also known as the stress hormone. And the next one is ethylene, which is a gaseous hormone. So it is the main function which is related to this is fruit ripening. So it helps in the fruit ripening. It plays role in apical dominance. It plays role in wound response. And it has an important role in thigmotropism. So thigmotropism is the tropism, that is the response to the mechanical stress, and it also has function in senescence. While auxin prevents abscission, that is the falling of leaves, prevents abscission and senescence. So this is the major summary of all the plant growth regulators that we discussed in the last class. This we need to remember by heart because most of the questions related to plant growth regulators are asked from this portion. The in today's lecture, what we will discuss is in today's lecture we will discuss the response of plants to various factors. Like majorly, we will discuss the response of light to response of plants to light, plant to light, response of plant to light. So this is known as phototropism. Photo is related to light, tropism is related to the response. So phototropism, as similarly plants also show thigmotropism. As I mentioned, thigmotropism means to the mechanical stimuli. Thigmo to the mechanical stimuli, mechanical stimuli, and gravitropism, that is to the gravity. That is gravity. This is mainly caused due to the presence of a component that is statoliths, which are present in cytoplasm. So they are granule-like structures that are present in the cytoplasm and that are responsible for gravitropism. Gravitropism, that is the effect for the gravity. So there are various types of tropism that plants show, and we will mainly discuss the phototropism, that is the response of plants to the light. So light is an especially important environmental factor in the lives of plants. We know that photosynthesis occurs in the presence of light only. So in addition to being required for photosynthesis, it triggers many key events, collectively known as photomorphogenesis. So the events, the morphological changes that occurs in the presence of light can be collectively called as the photomorphogenesis. That is the events in the plant growth and development which are mainly triggered by the presence or absence of the light is known as photomorphogenesis. So an example of photomorphogenesis, let me raise this. So example of photomorphogenesis can be explained with the help of taking an example of a potato. So it is the example includes a sprouting potato where we can see that this is a modified underground stem. So potatoes are modified underground stem which is known as tuber, right? So it has sprouted shoots as we can see in this figure. So it has sprouted shoots. However, this shoot resembles very less with the typical plant because we can see that in this the leaves are not grown well. So leaves just like the other stem where the leaves are grown in the expanded form, in this the leaves are not very expanded, they are not green, they are not adapted for photosynthesis as such. Morphological adaptations, so why the non-expanded leaves, they have a tall spindle stem. Why this is required? Because they are grown in the darkness, that is under the soil. So they are growing in the darkness under the soil. So therefore, in order to grow this shoot out of the soil, they require a lot of energy. Therefore, by these modifications, they are trying to conserve the energy. So a dark grown potato has tall, spindly stems and non-expanded leaves. These are the morphological adaptations that enable the shoots to penetrate the soil by conserving the energy. The roots are short, but there is a little need for water absorption because little water is lost by the shoots because obviously they are present inside the soil, under the soil. Therefore, there is a less uh, loss of water, and due to this, there is less requirement of the water absorption. So these are the morphological changes that are adaptations that the potato has undergone. So these morphological adaptations for growing in darkness are collectively referred to as etiolation. These morphological adaptations which the potato has undergone to grow in the darkness where there is no need for the expanded leaves, therefore non-expanded leaves are present. The stem is tall because it has to come out of the soil and it is spindle stems, and these are the adaptations which the potato undergo for growing in the darkness, and these are collectively referred to as etiolation. So when a shoot reaches the light, the plant undergoes profound changes. Now, when it reaches the light, we can see that after a week of exposure to the natural daylight, we can see that the leaves have expanded just like a normal plant. And this process is known as de-etiolation, that is informally known as greening. So now you can see that the greening of the plant has happened. Green leaves have arisen. So this process is known as de-etiolation, that is the reverse. So etiolation is removed. Etiolation is removed. De-etiolation. So in etiolation, the leaves are not expanded, the stem is long, but in this case, you can see that the potato plant begins to resemble the typical plant with broad green leaves, short sturdy stems, and long roots. While in the earlier case, the leaves were not broad, so they were non-expanded. Leaves were non-expanded. In the etiolated case, the stem was tall. So this I am writing the points of the etiolation. What was happening in the etiolation? When the plant was growing in the darkness. So the leaves were non-expanded, stem was tall, and roots were short. Roots were short because of the less requirement of the water absorption. So, but when the plant is exposed to the light, when the shoot reaches the light, the plant undergoes profound changes, which are collectively called as the de-etiolation, informally known as greenings. Now, stem elongation slows, leaves expand, roots elongate, and the shoot produces chlorophyll. So, in short, it begins to resemble a typical plant. And this transformation begins with the reception of the light, because obviously this response is this response is due to the presence of the light. So, light here is acting as a stimuli. So, when the plant is exposed to the light, the plant creates a response. And to respond, there must be some receptors that must be present, that must be present in order to detect this stimuli, right? So this transformation begins with the reception of the light by a specific pigment, which is known as phytochrome. So this reception of light by a specific pigment is known as, this pigment is the phytochrome. So this phytochrome responds to the light. So this phytochrome responds to the stimuli, which is the light, and due to this response, as a result of this response, de-etiolation happens. So phytochrome is the pigment that is present, that is a specific pigment that responds to the light. So plants do not only detect not only the presence of light signals, but so only the presence of light is not enough, but also their direction, from which direction it is getting the light, what is the intensity of the light, and what is the wavelength. So this is important, that even the wavelength. So in the visible light spectrum, we have VIBGYOR, that is violet, indigo, blue, green, yellow, orange, and red. The wavelength, this starts from 400 to 700 nanometer, right? So this wavelength, the wavelength also affect the growth of the plant. So plants detect not only the presence of the light is enough, but what kind of wavelength it is receiving, what is the intensity of the light, and what is the duration of the light, what is the direction of the light, is also a matter for the plant growth and development. So therefore, it is the wavelength of the light which is also a very important. So in case of photomorphogenesis we discussed, that is the changes due to the presence of the light. So action spectra reveal that the red and blue light. So red and blue light are very important. What is the wavelength of red light? So the red range from 600 to 700 nanometer. Blue light, the wavelength, that is lambda, is equal to 425 to 450 nanometer. It's 425 to 450 nanometers. So these two wavelengths are very important for the growth, and they are the most important colors for the plant. So red and blue. Red and blue are the most important light for the plant, most important colors for the plant. So we can see in this figure, this is the action spectrum, where we can see that, so when the white light when passes through the refractive prism, it undergoes dispersion. What is dispersion? When the white light passes through the refracting prism, this white light divides into its 7 components, which is violet, indigo, blue, green, yellow, orange, and red. So this is blue, this is indigo. Violet, indigo, blue, green, yellow, orange, and red. And when coleoptiles, that is when shoots are exposed to the light of various wavelengths, you can see that the bending can be observed slightly in violet and more in blue. Rest in all the colors, rest in the wavelength of all the colors, there was no bending. So we can see that the wavelengths, that is the violet light, induces slight curvature towards the light, and the blue light induces the most curvature. Therefore, we can say that every wavelength has a different effect. So the other colors do not induce any curvature. This phototropic bending towards the light is controlled by phototropin, which is a photoreceptor sensitive to the blue and violet light, particularly blue light. So we can see that in the blue light, there is a more curvature which is induced, more curvature. While in the remaining, we can see that there is no curvature. So no curvature. But in the blue, it is the more curvature. In the violet, still it is a slight curvature. So this is because of the photoreceptor, which is sensitive towards the blue and violet light and more towards the blue light. So this photoreceptor is the phototropin. So this phototropic bending towards the light is controlled by a receptor phototropin, and this receptor is specifically sensitive to the blue and violet light, particularly to the blue light, thereby showing more curvature effect in the blue light. So this example shows that not only the presence of light, but the intensity and the wavelength or the direction of the light is also important. So wavelength being different from each other, like the wavelength of the green is different from that of blue. Therefore, the effect is different based on the receptor pigment. So in the earlier case, in case of photomorphogenesis, I mentioned that the red and blue light are very important. But in this case, you might get confused that the red light is not inducing. So it depends on what action is being caused by the what photoreceptor pigment. For example, here, as I mentioned, that the phototropic bending is controlled by phototropin, which is a receptor pigment towards the blue and violet light. So this phototropin is responsible for the receptor for the bending, and this receptor is specifically sensitive to the blue and violet light, and therefore blue light is inducing the curvature. Now there are many experiments which were done in the past. So effect of light was also seen on the seed germination. So it was seen that whether the different light are able to cause or inhibit the seed germination or not. So dark period was taken as a control. The line below the figure of the seeds show the time period for which the period was maintained. For example, it was a complete dark period that was taken as the control. Therefore, there will be no germination due to the absence of the light. No germination due to absence of the light. So this is the control, absence of light. Then we have another set of experiments where we first expose the seeds with the red light and then we give a dark period. So the brief red light period induces the germination. While in the other set of experiment, when the seeds were first exposed to red, then to the far red, then there is no germination. In the next set of experiments, the seeds were first induced with the red light, then they were exposed for certain time with the far red light, then they were again exposed to the red light followed by a dark period. So it again shows germination. And if I again after the exposure to the red light, if I again expose it with the far red light, germination inhibits. So no germination. So what is happening in this experiment? From this experiment, we can conclude. What are the results? The results are that whenever we expose the plants with the red light, followed immediately by dark, then it causes germination. But if this exposure of the red light is then followed by the far red light, then it prevents the germination. But if far red light is then further exposed, then after this, if the plant is further exposed with the red light, then it again causes the germination. So there are two things that we get to know from this experiment. The first one is that germination is inhibited. So far red light inhibits. So we can say that far red light inhibits germination. Far red light inhibits germination. While if I talk about the red light causes the red light. So red light causes the germination. Red light promotes germination, causes germination. Also, what we can see is that the effect of the red and far red light are reversible. The effects of red and far red light are reversible. How can we say this? Because when the plants which are exposed to red light ones are then exposed to the far red light, then the effect of the far red light will be nullified by the far red light. And if again this far red light exposed plant is exposed to the red light, then again the plant shows germination. That is, the exposure of the far red light is nullified by the final exposure of the red light. So it is always the final exposure on which the result of the germination depends. Final exposure. So if the final exposure in the diagram is shown with the red light, then it causes germination, because we see that the effects of the red and far red light are reversible. And if the final exposure is shown by the far red light, then it will inhibit germination. This we need to remember. If the final exposure is shown by the red light, then the plant will show germination. But if it is the far red light, it will inhibit the germination. Right? So even if no matter how many times you expose it with red light, far red light, it is always the last exposure that will judge whether the plants will undergo germination, that will decide whether the plant will undergo germination or whether there will be no germination, because the effect of the red and far red light are reversible with respect to each other. So if in the question it is being shown that it is the red light which is the last exposure, we have to mark that yes, the plant can undergo germination. So this was the effect of light on seed germination. Now, very important topic, which is the photoperiodism. Photoperiodism, that is, the light and period defines the time, right? So the response of plants to the periods of day and night, that is the period, that is the time for which the light is exposed or the plant is kept in dark. So flowering in certain plants depend not only on a combination of light and dark exposure, but also their relative durations. That is, for how long the light or dark conditions are maintained. Right? So it not only depends on the presence or absence of light, but it also depends on how long we are exposing a certain plant to the light or how long we are keeping it in the dark. So this response of plants to periods of day and night is known as photoperiodism. So this response is known as photoperiodism, and it is very important in flowering plants, because there are many flowering plants that flower according to the photoperiodism. So their photoperiodic nature is different, that we will see that how we classify plants on the basis of this. So the phenomenon of photoperiodism was first discovered by Garner and Allard on Maryland mammoth, which is a mutant variety of tobacco, and Bloxy soybean. So it was firstly discovered by Garner and Allard, where they check for the growth of the flowers, they check for the flower, like the blooming of the flowers in the different time periods when the plant is exposed to different time periods of the land. So it was shown by Garner and Allard, and the plants in which it was shown was Maryland mammoth, which is a mutant variety of tobacco, and Biloxi soybean. So this is important that photoperiodism was first shown by Garner and Allard on Maryland mammoth, which is a mutant variety of tobacco, and Bloxy soya bean. So on the basis of this, Garner and Allard, classifies the plant into three categories. Classified the plant into three categories. First is short day plants, SDPs, short day plants. As the name suggests, they require a shorter duration of light. Long day plants, which require a longer duration of light. Long day plants, LDPs, and day neutral plants. That is, which are not affected. The plants in which it is not affected, the flowering is not affected by the length of the exposure of the light. So, on the basis of this, Garner and Allard were the one who first discovered phototropism. Photoperiodism. Phototropism was first observed by Francis and Darwin, right? Photoperiodism was first observed by Garner and Allard. So it was first discovered by Garner and Allard, and on the basis of this, he classified different plants into three categories: short day plants, long day plants, and day neutral plants, that is DNPs, that do not have any effect on the light. So short day plants, as the name suggests, that these plants require a light for the shorter time. So short day plants can be called as long night plants, right? That is short day and long night. So they require short days and long night. That is less time for light and more darkness. More time for dark. So these plants flower on exposure to photoperiod shorter than their critical day length. So whatever their critical day length is, these plants flower on exposure to photoperiod which is shorter than their critical day length. So critical day length is different. Critical day these plants flowers only when their photoperiod is shorter than their critical day length. So for example, if the critical day length is of 14 hours, so out of 24 hours, if it's a 14 hours daytime, then these plants will flower only when they will have an exposure which is less than this 14 hours. That is maybe for the 10 hours. Right? So these plants require a lesser light. These plants flower on exposure to photoperiod shorter than their critical day length. And in SDPs, the dark period is critical and must be continuous. This is very important. The plants were named as short day plants, long day plants, because when the Garner and Allard was actually observing the plants, they were observing it in the daytime. That it is the length for which the light is being exposed. But in general, in actual or in reality, what we know now is it is not the time for which it is not the length of the time for which the plant is exposed to light, but it is actually the period of darkness. And therefore, it is very important that the period of darkness must be continuous. Although even in today's plant physiology books, you will find that short day plants or the long day plants, but in actual, it should be long night plants, because it is the period of continuous darkness which is required. So it is the period of continuous darkness. It is the period of continuous darkness which is required. That is, long night is important. It is not only the short days, but actually the long night which is important. Because if it is interrupted by light, so if it is interrupted by light, the plant will not flower. Flowering will not happen. So it is the darkness, it is the period of darkness that we have to maintain. Therefore, continuous darkness is required. So it is very important. Then we should remember this short day plants as long night plants, because this will allow us to get the more better concept about this, that the long night plants, that is a period of continuous darkness, is important. If this period is interrupted by the presence of a light even for a very short period of time, then the flowering will stop, flowering will not take place. Thus SDP are also called as long night plants, and we should remember this. So if this dark period is interrupted even with a brief exposure of light, the SDP will not flower. Therefore, in actual sense, they should not be actually called as short day plants, but actually be called as long night plants, because it is the period of darkness that is more important. So, example of SDPs, you need to remember these examples. These are important. Example include tobacco, in which it was discovered, soybean, xanthium, chrysanthemum, dahlia, sugarcane, cosmos, rice, etc. So these are certain examples in which was short day, so examples of short day plants. So tobacco is important and soybean is also important, in which it was discovered. Next is long day plants. So long day plants will be short night plants. So these plants flower when they are exposed to photo period longer than their critical day length. So longer than their critical day length. For example, as I mentioned, let's say critical day length is 14 hours. So these will flower only when they will have the light exposure of more than 14 hours. So a brief exposure of to the light in the dark period stimulates flowering in the LDPs. So even a brief exposure to the light in the dark period stimulates flowering. Therefore, the light period is critical for LDPs. Gibberellin, as I mentioned in the starting of the lecture, that gibberellin plays an important role in flowering. Gibberellin can induce flowering in LDPs even under non-inductive photoperiod conditions. So it is important plant growth regulator that can induce flowering in the long day plants even under non-inductive photoperiod conditions. Example include henbane, spinach, sugar beets, wheat, larkspur, and barley. So these are certain examples of long day plants. And for the short day plants, it was tobacco, soybean, xanthium, chrysanthemum, dahlia, sugarcane, cosmos, rice, etc. Right? Then we have day neutral plants. So day neutral plants such as tomatoes, rice, dandelions are unaffected by photoperiod and flower when they reach a certain stage of maturity. So only when they reach a certain stage of maturity, regardless of the day length, they will flower. So they are called as day neutral plants. They do not have any effect, do not get influenced by the period of light, that how long the light is being exposed to. So this is the representation of SDPs and LDPs that I was trying to explain to you. So short day plants, which is also known as long night plants. So this bar shows a 24 hours time period, and the yellow thing shows the exposure to the light, and the gray thing shows the exposure to the dark. So the upper panel is for the short day plant. These three bars are for the short day plants, and the lower panel is for the long day plants. So in the short day plants, you can see that there are three conditions. This middle dotted line, break line, shows the critical period. So when in the short day plants, we know that in the short day plants, plant will flower when exposure to light is less than the critical period. Critical light period. So you can see here that in this case, the plant will not flower. But in this case, the plant will flower, while in this case also, the length of the time for which the light is being exposed is less. But since there is a hindrance that we cause due to the flash of the light, even the brief exposure of the flash of the light in the dark period causes the inhibits the flowering, inhibits flowering. It should be critical light period. When the exposure of light, critical light period. And this is the critical dark period which has been shown. This has been shown to make you remember that the short day plants are actually the long night plants in which the critical dark period is important for the flowering. So if up to that period the darkness is not maintained, the plant will not flower. So we can see that in this, the darkness is not maintained up to this, therefore there is no flowering. Even in this case, the darkness is maintained, therefore flowering happens. But in this case, there is a brief exposure of the light, and therefore there is a hindrance and inhibition of the flowering. And if I talk about the long day plants, which are the short night plants, flowers only if the night is shorter than a critical dark period. So in this, we can see that the plants flower only when there is a shorter dark period. But when there is a longer dark period than the critical dark period, the plant will not flower. You can see here. But again, when there is a exposure, when there is a brief exposure with a flash of light in the plant will flower, thereby inducing flowering, because they require a shorter critical dark period. Long day plants means shorter critical dark period. So in this, by the brief exposure of the flash of the light, we are actually what we are doing is we are shortening the critical dark period. So we are shortening the critical dark period, due to which there is flowering. So this is very, very important. It can be directly asked in the examination based on the figures. So we need to remember the concept of short day plants and long day plants, that what actually is happening. And I would suggest you to remember the alternative name for this, that is the short day plant is the long night plant and long day plant is the short night plant. And it is this night period which is very important for the flowering. So long night plant means that the period of darkness should be, that the period of darkness should at least be more than the critical dark period for the flowering. In the same way, for the long day plants, which is the short night plants, there is a shorter critical dark period which is required. So the period of darkness should be less than this critical dark period. And even within this, if the darkness remains, suppose this critical dark period is of 14 hours. And within this 14 hours, even for a minute, or let's say for an hour, I expose the plant with the light. I expose the plant with light. So what will happen in short day plants? In short day plants or in the long night plants, flowering will stop. But what will happen in the long day plants? Due to the exposure of the light in this critical dark period, the plant will get the exposure to the light and it will interrupt the long dark period, thereby inducing the flowering. So what is responsible for this is the phytochrome. So these are the receptors that are present, or the pigments which are responsible for this. So a light sensitive pigment, which is a phytochrome, which is a chromoprotein, is responsible for flowering induction, and it is present in leaves. This is important. It is present in leaves. And it exists in two forms, that is the phytochrome red, which is represented by PR, and PFR, which is the phytochrome far red. So both forms of phytochromes are photochemically interchangeable with each other. That is, they interchange with each other in the presence of light. Photochemically interchangeable means in the presence of light. So in the presence of light, red, that is PR, can get converted into PFR, or PFR get converted into PR. And it is the fluoregin. This is important. This is a hypothetical hormone. Hypothetical means which has not yet been identified, or it is just the hypothetical hormone which is not yet isolated and proved to be. So it's a hypothetical hormone, florigen, which migrates from leaves. As I mentioned, that it is phytochrome is present, responsible for flowering induction, and is present in leaves. So it is the leaves in which the hypothetical hormone florigen is also present, that migrates from leaf to shoot apexes for the induction of flowering. So what happens is that the phytochrome red, in the presence of the red light, undergoes the conversion to the phytochrome far red, and phytochrome far red, in the presence of the far red light, undergoes conversion to the PR, that is the phytochrome red. But this conversion, that is the conversion of phytochrome red to the PFR, is faster, but the conversion of PFR to PR is very slow conversion in darkness. And this PFR leads to the enzymatic destruction that may result in the seed germination. It also inhibition of vertical growth and stimulation of branch, setting internal clocks, and control of flowerings. That is, the long day periods or the short day periods are being influenced by this phytochrome far red. So this is the molecular switching mechanism. The absorption of red light causes PR to change to the PFR, and the absorption of PFR to the in the phytochrome far red light causes its conversion to the phytochrome red. Please do not get confused that since it is phytochrome red, so it will be synthesized in the presence of PR. But actually, phytochrome red will undergo the conversion to the PFR in the presence of red light, and it is a very fast conversion in the presence of the light. But PFR slowly converts during the darkness in the PR. So far red light reverses this conversion. That is, it reverses the conversion from PR to PFR. So in the presence of far red light, PFR will be converted to PR. And remember this hormone, hypothetical hormone name, which is florigen. So this was about the photoperiodism, and the pigment which was responsible for this is the light sensitive pigment, which is the phytochrome. The next is the vernalization or yellowization or springification, that is the spring. That is flowering. So flowering in certain plants is not only affected by the duration of the light, but in certain plants it is also affected by the temperature to which the seeds are previously exposed. For example, if I talk about the wheat, we know that for the flowering in the wheat, we require its exposure to the long period of lower temperatures. So some plants bloom after a single exposure to the photo period required for the flowering. However, there are other species that need several successive days of appropriate photo period. So in certain, a single exposure of the photo period is enough for flowering, while in others, there are several successive days which are required for the flowering. Still, there are others that respond to a photo period only if they have been previously exposed to some other environmental stimulus, such as a period of hold. So this is the vernalization. This is where the vernalization come into the picture. So other plants, there are some plants that respond to a photo period only if they have been previously exposed to some other environmental stimulus. Until or unless we do not expose the plants to this environmental stimulus, irrespective of whatever the periodism we are following, the plant will not flower. So this is very important step in the flowering of the plants, where in certain plants, these plants respond to photo period only if they have been previously exposed to some other environmental stimulus, such as a period of hold. So this is known as vernalization. For example, the winter wheat, for example, will not flower unless it has been exposed to several weeks of temperature below 10 degrees centigrade. In certain books, it has been mentioned 5 degrees, but the point to remember here is that several weeks of lower temperature is required for the flowering. The use of pre-treatment with cold to induce flowering is called the vernalization. So this use of cold pre-treatment to induce flowering is called as the vernalization, and this has been arrived from the Latin word which is spring. Therefore, it is also known as springification. because we are inducing flowering, springification. Several weeks after winter, wheat is vernalized. A photoperiod with long days, that is the short nights, induces the flowering. So it is only after the vernalization, that is after the exposure to the cold, then the photoperiod comes into the picture in certain plants. So this is known as the vernalization. So this is the effect of vernalization, where a slightly germinated seed is chilled for a month at 5 degrees Celsius, planted at ordinary temperature. So now, which was a previously chilled at 5 degrees Celsius, is planted at ordinary temperature. So this is a vernalized plant. Vernalized plant. So this vernalized plant shows flowering because already the vernalization has happened. But if no chilling has been done and we plant it at ordinary temperature, then the plant is non-vernalized and there will be no flowering. There will be no flowering. So flowering will not happen, while in this case the flowering will happen because of the vernalization. Vernalization. Right? So this is important. This is the effect of vernalization, where if certain plants that require a certain period of lower temperatures is not exposed to that lower temperature, they will not flower. So what are the requirements and mechanism of vernalization? Firstly, it requires actively dividing cells. It requires a period of low temperature treatment. It requires aerobic respiration, optimum hydration, optimum nutrition. These things are important, but the very important thing is period of low temperature treatment. Mechanism is that after the perception of a suitable low temperature period for a successive time, a hypothetical hormone called vernalin. So in this, we talk about a hypothetical hormone, florigin, which is responsible in the photoperiodism, but in vernalization, it is the hypothetical hormone vernalin is produced. So vernalization involves the hormone vernalin. Photoperiodism, that was responsible for flowering. The hormone was florigin. Flori is related to flowers, florigin. The hypothetical hormone was florigin. For vernalization, it is vernalin. So only after this vernalin is produced, then comes the picture of the photoperiodism into the picture. So examples include that the process of vernalization is significantly employed in food plants like wheat, barley, rice, etc. In biennial plants that have two seasons like cabbage, sugar beet, etc. And perennial that comes throughout the year, chrysanthemum. So some important food plants, for example, wheat, barley, rye, have two kinds of varieties, spring varieties and winter varieties. Now, what is the difference between spring varieties? So spring varieties are normally planted in the spring. As the name suggests, they are planted in the spring. Spring where we do its plantation. They are planted in the spring and produce and come to flower and produce mature grains before the end of growing season. So they come to flower and they come to flower and produce mature. They come to flower and produce. They come to flower and produce mature grain before the end of growing season. Before the end of growing season. While if I talk about the winter varieties, as the name suggests, they are planted in the late autumn, that is before the winter. They are planted in the late autumn. They germinate and over winter come out as small seedlings. They germinate and over winter they come out as small seedlings, which resume growth in spring, and are harvested midsummer. So this is important. So winter varieties are actually harvested midsummer and spring varieties are planted in the spring. They come to flower and produce mature grain before the end of the growing season. Winter varieties are planted in the late autumn and they produce small seedling after the exposure of the winter. In the exposure of the winter, helps them in producing the small seedling, and they resume the growth in spring and are harvested in the midsummer. Winter varieties, however, if planted in the spring, would normally fail to grow or produce the mature grains within a span of flowering season. So, some might argue that the winter varieties can be sown directly in the spring. So, winter varieties, if they are planted in the spring, they would not be able to undergo flowering. Similarly, in the biennial plants, if I talk about the biennial plants, so biennial plants have two seasons. Biennial, bi means two. So biennial plants, so in the first season, what they do, so they have two seasons, first season and second season. In the first season, they germinate, they resume growth, they resume growth, they resume growth, and ultimately attain maturity In second season, they flower. The flower then form the fruit, which then sets seeds. Because fruits are formed, fruits will form the seeds, and the plant undergoes senescence, senescence, that is the period of aging, senescence, and the plant ultimately die. So these are the two seasons. In the first season, what happens? They germinate, they resume the growth and attain maturity. After attaining the maturity in the second season, these plants produce fruits, they set seeds, they undergo senescence and die. Right? So, what is the importance of vernalization? Vernalization gives adequate time for the plants to get mature and also prevents prematuring during the growing season. So, during the growing season, it prevents prematuring. It gives the plants to get mature at a specific time, and it gives the adequate time for the plants to get mature before growing. It reduces the vegetative phase and hastens the reproductive phase. Vernalization increases the plant's growth and yield. Vernalization makes the plant more adaptable by allowing them to grow in regions where they usually don't grow. And it removes wrinkles on the kernels of the triticale. Triticale is the wheat. It is not only applicable for growing temperate plants but also some tropical plants like wheat, rice, millets, etc. The next topic is seed dormancy. So seed dormancy refers to the seed failure, that is when germination is not able to take place. Germination does not happen. Right? So seed dormancy refers to the seed's failure of a viable seed to germinate even when there are favorable environmental conditions. So there is dormancy when but there are certain dormancy when the seeds undergo dormancy due to unfavorable condition, but as a favorable condition arise, it undergoes germination. But there are certain situations where there is the situation where the seed undergoes dormancy and it fails to undergo germination even when the favorable environmental conditions are given. Such seeds, what we expect from these seeds is that they are understood to be undergoing a period of dormancy which is not controlled by external environment. So obviously in such kind of situation, the external environment is not affecting the dormancy, but it is the internal endogenous control or conditions within the seed itself which is preventing the germination. So even after the reversal of the favorable conditions, if the seed is not undergoing germination, then it is not the external environment which is responsible. It must be some endogenous control or conditions within the seed itself. What are the reasons? So the reason for seed dormancy is impermeable and hard seed coat, therefore not allowing the water to enter. Presence of chemical inhibitors such as abscisic acid, we know, is an inhibitor. Phenolic acids, para-ascorbic acids, etc. Or the embryo is immature. So how can we break seed dormancy? Seed dormancy can be break by two ways, natural and artificial. So there are certain natural ways by which the seed dormancy is broken. First is the action of sunlight. So it breaks down the germination inhibitors like ABA, right, allowing the growth. The second is the action of heat. Again, the exposure to heat breaks down the germination inhibitors. Passage through the digestive tract. So there are certain seeds which when passes or digested by the animals, then they undergo germination. So when seed passes through the digestive tract, Hard seed coats become soft due to the action of enzymes. Due to the action of various enzymes, the seed coat becomes soft, and also it this enzymatic action also eliminates the inhibitors, thereby allowing the growth. And by microbial action, that again release certain enzymes that may soft the hard coat. There are certain artificial ways also. So first is stratification. Physiological dormancy is broken by the exposure to cold temperatures. Scarification, it is the breaking of dormancy by nicking the seed coat with the sharp knife, where we just nick the seed coat with the help of the sharp knife, or we use the sandpaper, or by vigorous shaking. The third one is water. So soaking seeds in the water will make it soft and will soften the hard seed coat enough to allow the moisture inside so that the seed can germinate. Or the application of certain chemicals. For example, we know that gibberellic acid is responsible for the seed germination. So gibberellic acid or the application of the nitrates are often used to break down the seed dormancy and to induce the germination. So this was the last lecture about the plant growth regulator and the plant growth and development. And in this, we discussed about the photoperiodism, vernalization, and seed dormancy, which are important features of the plants. And it shows that how the growth of the plant is affected by the presence of the external environment, like by the presence of the light or the temperature, etc. So, thank you for attending it.